Really needs no introduction. He started at the university in 1976. He is Regents Professor Fred Allendorf. And I have known him for a long time. He's lectured in my classes. He, as you guys know, he's into evolution, he's into population genetics, and he's into conservation biology big time. And he got his bachelor's from Penn State, and he got his master's and PhD from the University of Washington. And he has distinguished himself in many fields, but he really likes to work on salamonid fish. The salmon, which don't include all the things called salmon, right?
No, they do.
They do, okay. Yeah. The evolution of genetics.
You don't call salmon.
And he has written some books. He's written, his most recent book is on conservation genetics book, co-authored on conservation and genetics and populations. And he teaches conservation genetics and a popular course called Population Genetics Seminar. And I've had my students take that. And he's got a lot of distinguishing characteristics as we just look at him, but he also has some distinguishing characteristics on paper, and he is a NATO NSF fellow. He's a member of the AAAS, which is a big deal. That's the American Academy of Science. And he's past president of the Genetics Association, been a Fulbright scholar, and to say more, he has a passion for science. And he's going to be speaking to us about the same topic, his passion for science. Thank you.
Thanks, George, for the introduction, and thanks for inviting me to talk. And so when George invited me, he said, I don't want you to give one of your usual boring lectures. I'd like you to talk about how did you become a scientist? And so that made me think, how did I become a scientist? And also, it's much more of a challenge for me to talk to a group like this than it is to talk about genetic drift or natural selection. But I'll try to entertain you for about 15 or 20 minutes. So how I became a scientist, I remember I was a chemical engineer major, and if we're going to consider engineering, not science, I took a course in anthropology, and it was physical anthropology, and so part of the course was talking about genetics and population genetics, and I'd never heard of that before. And I remember going home that night and lying in bed and thinking, that's what I want to do for the rest of my life. So that one lecture, that one course has totally switched everything that I did, and I became a geneticist, and became fascinated with genetic variation in humans, salmonids, and other species as well. And when George asked me this, I began asking other people, my colleagues, about how they became a scientist. And Winsor Lau, a good friend of mine in biology, he had a parallel experience. He was a political science major, and he took a course in vertebrate evolution. And he said when he took that course, he decided he wanted to be a biologist. And I have one other example of that, somebody else that you all know that had a similar kind of experience. So, I put passion in my title because I think it is important to have passion to be a good scientist. And I guess it was that one lecture in genetics that for some reason I recognized the passion that I had for genetics, partially because you don't have to memorize anything in genetics. It's just a matter of understanding things. I've never been able to memorize things very well. And so I've been here, as George said, at the University of Montana since 1976. And I've spent a lot of time at Victoria University of Wellington in New Zealand, and I currently have a 20% appointment there. So this is a great time to be a geneticist, all the amazing things that are happening right now. And so this is Craig Ventier, the first complete human genome to be sequenced. It's really interesting. He was he was the driver behind sequencing it, and he had to pick somebody, and so he chose himself. And so the complete 5 billion base pairs of Craig Ventier's sequence you can find on the internet if you would like to. And who do you think the second person to have the complete genome was?
Bill Gates.
Good guess. I'm sure, people today happens to be my birthday. And I told my lab, said, what do you want for your birthday? And I said, I want my complete genome. And if I were Bill Gates, I would have it. But that's a good guess, but who else do you think it could be?
Craig's wife.
Craig. This isn't obvious, is it? James Watson. Watson and Crick. And so the second genome was James Watson, but, and this is really interesting, almost. So he did not allow the public information of what his DNA sequence was as the gene primarily responsible for Alzheimer's disease. So the whole rest of his genome was available, but not that particular gene. And just like myself and Winsor Lau, it was one course, one lecture that really made the difference for James Watson. He wanted to be Dick Hutto when he was an undergraduate student and studied birds. He had a session for birds, but one course from Sewell Wright at the University of Chicago, and he decided he wanted to study genetics. And probably only a handful of people in here ever heard of Sewell Wright. He is one of the top population geneticists, if not the top population geneticist of the 20th century. But Sewell Wright is infamous for being the most boring lecturer in the world. And I have some personal experience with this. He came and gave a seminar over at Bozeman in the mid-1980s, and they told him he had 50 minutes to talk, and 2 hours later, they literally had to drag him off the stage and say, you can finish the talk tonight, Sewell, at the banquet. So he was talking about his whole career, and he got, which started in 1920, he was about up to 1932 when they finished. But he was, and this was not an unusual experience. So I think the fact that Watson, it wasn't Sewell Wright's lecture that made Jim Watson want to become a geneticist. There had to be something there in the material. And I think this is one of the points I'm trying to make, is that this passion for whatever we do in science is really important, and it's sort of serendipity when we find that, how we discover that passion. And so Watson is not one of my favorite scientists, but he's done a lot of interesting things. He's gotten in trouble recently for a number of things, but he wrote an autobiography a few years ago, and there are his rules for success as a scientist. So the first one is avoid dumb people. And take risks and be prepared to get into trouble. Have a fallback, someone who will save you when you're in deep shit. I don't know about this first. I like the fourth one, have fun and stay connected. Never do anything that bores you. And 5, if you can't stand to be with your real peers, get out of science. I'm not sure what he's talking about there, but there's another rule of Watson's that I really do like, and somehow it didn't make his top 5. And I read this a long time ago, actually, as one of my first sabbaticals. It's necessary to be slightly underemployed if you are to do something significant. And I think, and this is true in my experience, when there are, so as being a faculty member, as many of you are, and some of you are likely to become, you're always busy. You're really, as Scott Samuels sort of talked about, he now lives vicariously through people working in his lab. You don't really have a lot of time to think about science as you did when we were graduate students. So I think it really is somehow arranging our lives so we really are slightly underemployed, really would be a valuable way to be able to do better science. And so we've gone way beyond Craig Ventier and Watson. This paper was published a few weeks ago, and this was a family in Seattle that both children had 2 genetic diseases. And so they were interested in finding out, they didn't know what genes were responsible for those diseases. And this is just amazing. So what they did was they completely sequenced both parents and both children. The whole genome of all of these. And I think they said the cost was about $40,000 a piece for each of the 4 individuals. And this is just mind-blowing for somebody like myself to think that this kind of information is being available. And it doesn't take a lot of imagination to think that in a few years, when you go into your doctor, I think what your doctor's going to do, they're not going to completely sequence your whole genome, but they are going to have little chips which can look at maybe the 96,000 key genes which are known to affect human clinical problems, and they will probably genotype you at all of those markers and then base their diagnosis and their treatment upon your complete genotype. And when I started, I was really lucky for lots of reasons. I was really lucky to come to the University of Montana. I've really loved it here. I've been here for a long time. I started in population genetics at a really ideal time, because it was in the late 1960s that for the first time people were able to use well, it wasn't called molecular biology then, were able to use biochemistry to look at genetic variation in natural populations. So before 1970, if you were a geneticist, you worked with fruit flies, or you worked with mice, or you worked with one or two plant species. But at 1970, the biochemical and the molecular techniques came along so that you could look at genetic variation in everything. And I've lived through that entire revolution, from proteins, the mitochondrial DNA, to microsatellites, to being able to sequence complete genomes. So it's really been an amazing time over these last, well, I started graduate school in 1971. And as George mentioned, my interest is evolution and genetics. And there's a consortium of geneticists, of which I am one out of about 27,000, that have proposed or trying to get the money to sequence the complete genomes of 10,000 vertebrates. And the idea is that within 5 years, you may be able to do a complete genome for just a few $100s. And so what the process now is trying to get the money and trying to get the samples, and so that we can go out and really look at the genomes of 10,000 different species. And from an evolutionary perspective, it's just amazing what kinds of insights would come from this. When you go back, if you read the first papers on the human genome, sometimes when I teach genetics and evolution, which I'm going to be talking about in a minute, molecular biologists don't want to hear about evolution. They just want to hear about molecular biology. But you really can't understand patterns that you see in genetics without understanding evolution. And the first paper on the human genome, most of the writing in there, most of the interpretation was from evolutionary biology. And so I taught genetics and evolution. Well, it used to be just evolution, but since I came here in 1976, and one of the reasons that genetics appeals to me is that it is the same principles from things that Scott works on, and E. coli, up to elephants, if you're interested in conservation of elephants. And I've thought about my time here at the University of Montana. I estimate that I probably taught about 5,000 students and 5,000 undergraduate students in evolution since I've been here. And I think it's one of the things that when I go around the country and give seminars, people often come up to me and say, oh, I took a course from you in 1978 or 1982, and I know what course it was, but it was 200 people. I usually don't remember them. And Theodosius Dobzhansky, one of the most famous population geneticists of the 20th century, along with Sewell Wright, made the quote that I think probably everybody in here has heard of, that nothing in biology makes sense except in the light of evolution. And it's becoming more and more appreciated that this is true. And one of my favorite examples that I always talk about to my undergraduate students is, why does human feces smell like shit? Does anybody know?
I think when I took your class in about 1982, wasn't it? So we didn't eat it? Is that?
Yeah, well, that's no fair. Yeah, exactly. Because, I mean, there's nothing shitty about shit intrinsically in terms of the smell. Because, I mean, dogs love to eat human shit, so it must smell good to them. So the reason that it smells shitty to us is because if we play with it and we eat it, we're likely to get sick from transmitting disease from one person to the other. So it's the process of evolution. Those individuals who did think that shit smelled good didn't live to reproduce. So they were removed from the gene pool. And since I'm talking to Sigma Xi, one of the things I thought about is, so I looked up, what is Sigma Xi all about? I've been a member of Sigma Xi off and on several times throughout my career. And George mentioned, I can't remember what he called it, but this is the mission, according to the webpage, to enhance the health of the research enterprise, foster integrity in science. And I guess what I want to focus on is promote the public's understanding of science for the purpose of improving the human condition. And this brings up the idea of basic versus applied science. And I guess I've always tried to get throughout my career work somewhere right in the knife edge between those two. And so we now have this idea that National Science Foundation as funding basic research. And people that don't already have their PhD, not other people that know the answer. Does anybody know where the National Science Foundation came from? What was the organization called before it was called the National Science Foundation? So it was in the about 1948 that NSF came into existence. Does anybody know what it was, sort of what its ancestor was before it became NSF? It was the Office of Naval Research. And so it was funded so that we could build military weapons. And so it has, even though we think of it as basic science, it really had an unusual history in terms of where the funding came from. And I was a program officer at NSF in 1989 and 90, and we were taught then about the scientific onion. And the center of the onion was physics, the next layer of the onion was chemistry, and this sort of represented how much money went into the different layers of that. Then biology, then social sciences, and other ones. And so when certain presidents came along, so when Reagan came along, he tried to peel the onion and remove social sciences from the onion. But as I said, I've tried to work in my career in this place between basic and applied, and my interests have really made that easy. So I've worked in the area of conservation and genetics of populations, trying to apply what we know about basic population and evolutionary genetics to issues in conservation. And I'm fortunate in that I think some of the most interesting basic questions are also the same questions that we need to know the answers to apply genetics to conservation issues. And one of the things I've always thought is that laughing is good. My wife encourages me to laugh a lot. So when I teach conservation genetics, I try to use conservation genetics for the joke of the day. And so this is a Canadian fur seal, and the Canadian fur seal went into a bar in Halifax, and the bartender asked him what he wanted. And he said, anything but a Canadian club. So in trying to apply genetics to issues, one of the issues that we've been involved in a lot is Endangered Species Act issues. So if the other day there was an article about Fisher in the Missoulian. It was genetic data that had made them reconsider whether or not Fisher should be listed. Wolves, the big issue in wolves is whether or not wolves are genetically isolated. Grizzly bears, one of the big issues, are grizzly bears in Yellowstone. Well, they are isolated from grizzly bears in Glacier and the Northern Continental Divide ecosystem. But the question is that isolation a problem? So are there enough grizzly bears in the Yellowstone ecosystem so that they're not going to suffer from inbreeding depression, which may cause them to be less likely to persist in the long run? And so we've done a bunch of models, and we've collected a bunch of data, and we've made the argument that they are too small and too isolated, and there needs to be some genetic exchange between Yellowstone and Glacier Park. And this is part of the recovery, not the recovery, but the delisting plan is to move bears back and forth. But you can imagine why Chris Servine doesn't want to go around taking grizzly bears from Glacier Park and putting them into Yellowstone. Because first off, it's difficult to deal with grizzly bears. And if you can imagine if the grizzly bear that they moved into Yellowstone Park attacked the tourists, the amount of money the federal government would be sued is beyond belief. So people have worked up with alternatives so that we can get the genes from Glacier into Yellowstone without getting the bears into Yellowstone. And this has been one attempt of trying to do that. So if you can't read this, this is the grizzly bear artificial insemination gene. And this is a joke postcard, but I really think it's true also. I think that it would be a great way if we could just get some sperm into some Yellowstone grizzly bears, then we really could alleviate this problem. And one of the things that turns out from Sewell Wright is that it doesn't take much exchange between populations to alleviate the problems of inbreeding depression. So if we can just get a few bears, a few genes from Glacier and Yellowstone, that would go a long way to solving the problem. So I just wanted to end with, I didn't know what to call these. They really weren't. They're just sort of random thoughts that came into my head that I wanted to leave people who are embarking on their scientific career. So the first one is love it or leave it. Understand something really well, ask good questions, collaborate with others, and enjoy yourself. So I've been in science a long time. The one thing that is clear to me that if you really don't have a passion for what you're doing in science, you're not going to enjoy it and you're not going to be very successful at it. It really is a lot of hard work, and if you don't enjoy doing that hard work, you probably should be doing something else. And sometimes, there's this choice between sort of being broadly trained and knowing a lot and being very narrowly trained. And I think all the efforts now talking about multidisciplinary and other things, I think we're pushing people to become broader and to know more about different things. And I think there's a real danger in that. And in my experience, as Richard Feynman said, I really love this quote. There's a lot of great quotes from Feynman. If we really understand a glass of wine, we're not only going to see the entire universe, but we're really going to understand the entire universe. So I think that it's really important to be interested in lots of things. Yes, to have broad training, but it's also really important to know some aspect of your science really well. And if you're an evolutionary biologist, I mean, there are certain things that you really need to be an expert in, simply because by really understanding, so in population genetics, the concept of effective population size, so is the concept of trying to explain it. There's also a quote from Feynman that said, they asked him to explain the work for his Nobel Prize to the general public, and he said, if I could do that, they wouldn't have given me a Nobel Prize for it. So I won't try to go there, but I think it's really, there's a danger in being too broad. I think it's really important to really know something really well and dig down into that, because when you dig down into it, you'll see, just like Feynman pointed out, whatever that is, it's going to be connected to the whole rest of the universe, and to really understand that small piece, you have to understand the whole picture. Ask good questions. I remember when I was a graduate student, the most intimidating member of my committee, every time I went in to talk with him, he always would look at me and say, "What's the question?" And if I couldn't tell him what the question was, he asked me to leave his office. And Einstein, I love this quote, it's asking the really hard questions. So I think the people that turn out to be successful scientists, and by successful I mean being happy in what they're doing, to be recognized by others, are those people that really choose the key questions. So if you're be a graduate student, get your PhD in something, rather than just pick something that you think you can publish 3 or 4 papers and get your degree, you really should pick what is a really hard and what you think is a really important problem in your field. Collaborate with others. I think this is really important. One of the most enjoyable things about my career is collaborating with lots of people all over the world. And I would say teaching is sort of a collaboration. And I've always loved this quote from Ari's wife, that when we teach, we really teach ourselves. And if the students understand something, that's fine, but it's not really very likely. And the most important thing is to really enjoy what you're doing. And I just have some slides of me enjoying what I do. So this is some field work we did in Alaska a few years ago on pink salmon. So we went out to, these are all pink salmon in this stream, which is up in Resurrection Bay in Alaska. And we were collecting fish, male pink salmon on top, female on the bottom. And we were taking samples from them to get their DNA, to get their proteins, and we also took eggs and sperm and did a number of experimental matings. It was a blast. So I spent a lot of time traveling with my wife, who's sitting here, Michael, and so this is a class we did with Ken Dial's course. We went to Africa, had an amazing time in Africa. Imagine, so I went as a faculty member, imagine getting paid to go to Africa and just spend 2 weeks on safari. My wife and I spent a lot of time in the last few years in New Zealand and Australia, and I have a lot of collaborations down there, and it's been really, and one of the nice things about conservation genetics is that the problems are the same, and so I've spent a lot of time working in New Zealand and Australia on the same kinds of problems I work on here, and it's been very rewarding as a scientist and also a heck of a lot of fun. And anybody have any idea where that picture was taken? Recognize Ronald McDonald? Anyone?
Japan?
Good guess. Good guess. Right, obviously Asia. But you know, if you had to think, no, it's not China or not Japan. Thailand, yeah. Thailand's sort of the ultimate capitalist. And so we were on a trip in Thailand and came across that. We weren't going into the McDonald's, but we parked outside the McDonald's. And we spent some time out in the field. These are Asian elephants, so I've been lucky enough to see both Asian and African elephants out in the wild. And I've done, in conservation, I've done some things which are sort of non-traditional. So this is a paper that I published in conservation biology, trying to sort of incorporate some ideas of Zen into conservation. Which is why I'm so fascinated by the Ronald McDonald Gaucho. And so this is my last slide. It didn't fit in with anything else, but I thought it was really nice, by Max Planck. And so if you're having trouble getting your colleagues to understand what you're talking about, for a long time scientists have recognized it's really difficult to get people to change their view of the world. The way that new ideas really become accepted is by when the next generation of people that became members today grow up, then these ideas will be accepted. So I'd be happy to answer any questions, or happy not to answer questions.
Somebody get the lights, please.
If I'd known there had been a plan in here that took genetics and evolution 20 years ago.
Well, Fred, what do you say? One student asked me some years ago, they said, well, you became a professor, and you've gone up through the ranks from assistant all the way to full professor and the prestigious Regents professor. Well, they said, why would somebody want to do this? Why would they want to go and study for 8, 9, 10 years and then have to compete in this terribly competitive situation for jobs that only a few can get, and then have to publish very, very hard and all this stuff? Why do you do it, they ask. And,
Well, this is trite, but it's true. It's the journey and not the destination. So I never thought about that. I mean, I just was always doing what I enjoyed doing.
yeah.
And so I never went to graduate school to become a professor, or it was just amazing that people actually paid me money to do stuff that was so much fun. I remember when I just started graduate school and I started doing experimental crosses with steelhead trout in the state of Washington. It was amazing to me that I could go and grab a female and a male and put the sperm and eggs together and come back 3 weeks later and there were baby fish inside the eggs. It was amazing. So I never really thought about where I was going. I just enjoyed what I was doing.
With your work with the genome sequencing and trying to get 10,000 species done, was that featured in National Geographic this month? They did a thing about like barcoding everything. People that work in tropical rainforests, it's really hard to tell, like genetic variation. Is this butterfly really a different species or does it just look different?
That's similar but different. And so that is so barcoding means just looking at about 1,000 base pairs in the mitochondria. So most of our DNA is in the nucleus and then some is in the mitochondria. The mitochondrial genes are very conserved, and so you can look at very different species and look at the same genes. And so the idea there is just to look at the sequences, and barcode is exactly what they are. Some of us think that that's not really interesting in science because all they're trying to do is identify individuals. And I think it's really important for conservation. But the genome sequencing will be able to understand, so the first genome is looking at humans and chimps. You can look at humans and chimps, you can say, okay, which regions are humans and chimps really different? And those must be the regions in which humans diverged in the last 3 million years from chimpanzees. So it's trying to understand the process of evolution, where barcoding is just trying to use DNA as a marker. Chris?
Right, given the amazing changes you've seen since 1970, could I tempt you to look ahead a little bit and imagine some of the things in the next 30 years that might be astounding? For example, some questions we might be able to answer that we have no hope of answering now.
I'm not underemployed enough right now. But I'm not very good at that. I don't think I'm very good at that kind of stuff, but I think that's what we should. I'm not worried about 30 years. I wish I could figure it out 5 years. As a population geneticist, the machines that we use cost hundreds of thousands of dollars. Do you go out and get a machine for $500,000 and 2 years later realize that it's no longer useful? And so I guess the time frame that most of my colleagues and I are thinking about is where are we going to be in 5 years? And I think the consensus is it is whole genome sequencing and not just looking at bits and pieces of the genome.
Yeah, I'm just curious point. Our colleagues over in computer science have been telling me that the rate at which we're going to get genetic information from so many organisms means in 2 to 3 years we'll run out of storage space to usefully transport and work with that data. So there actually needs some computer science solutions to start working on cool questions in 5 years.
And that's completely true because it's not like when you sequence a genome, you get the 5 billion base pairs, but you have to sequence it 70 times because a lot of times there's mistakes. And so you got to do it 70 times and then look at it to make sure and then try to correct your mistakes. So the amount of the amount of data is just amazing, and when we start looking at thousands or tens of thousands of human genomes, 10,000 vertebrates, in 30 years, sometimes I think that but probably everybody who's been in the field 35 years thinks that. There really aren't any interesting, well, there are no more data out there. So when I was a grad student, first we had proteins, and so we kept getting more and more data, and we kept trying to get better machines to get more data. But now you have the complete genome. I mean, what's the next step? There's nothing. So I sometimes feel like it would discouraged about young people in evolutionary genetics right now. But I think that's just my limitations, because I think the questions are just going to change. It's not going to be collecting the data. It's going to be trying to be using the data and try to come up what really are the questions that we need to be asking.
Is the primary goal of your 10,000 species to just have a definitive trail of evolution, or what is the goal of that?
No, so it's not just reconstructing relationships, because we don't need the whole genome to do that. It really is trying to understand what changes are taking place, what in the DNA to bring about functional differences. So one question would be, so cats and dogs. So it's amazing to me, dogs and cats were domesticated about the same time. But look at the amazing phenotypic variability in dogs and very little phenotypic variability in cats. So cats, they're pretty much all the same size, and there's some that are more hairy, a little bit larger, but there's really not much variability in cats compared to dogs. So by looking at whole genomes, it would be nice to know, well, why is it possible for dogs to very quickly evolve all these different forms where it hasn't been possible in cats? So it's asking, and like the human and chimp question that I had, trying to identify those changes which are really crucial for major evolutionary advances that we see. So flatfish, so flounders turn over and their eyes migrate to the top of their head. So, by looking at a bunch of pleuronectids in that same family, when did that change take place in evolution? How many genes were involved? What kinds of changes took place? So really it's trying to take the genetic information and understand how it's affecting development. Other questions?
Where are we going to find the heart of science in the next 30 years?
Well, I had a friend who was an evolutionary biologist who just died in his early 80s, University of Hawaii. He worked on fruit flies his whole life, and he died a discouraged man. What he said was, here I've spent my whole life studying evolution, and half the people in my country don't even accept that evolution is a process that is taking place. So I guess where the heart would be trying to get other people in our society and other societies to be able to evaluate information from the world in a scientific manner. 30% of the people in the U.S. think that Barack Obama wasn't born in the U.S. I mean, it's just amazing that people are so capable of ignoring what seem to be fairly straightforward facts.
Well, this is really astounding. I remember in a poll that was done about what people's perception of science would be, when they had a choice to choose from, they almost inevitably chose to define science as the structure, the organizations, the institutions, the machines, the institutions of great research, not a way of inquiry.
Yeah, well, George I. Bush, he had that famous quote about science, the fact thing. But I always often make the point to my students, who was the greatest scientist of the 20th century? Most people say Albert Einstein. Albert Einstein never found any facts. All Albert Einstein did was think about things and try to present hypotheses. So yeah, I think if we could, people have this misconception of what science is, I agree.
Any other questions? Let's thank him again.